We were getting pretty good at packaging proteins into vaults, and we were expanding our understanding of the chemical properties of vaults. It was tempting to imagine that vaults might someday be engineered to deliver some therapeutic to humans. Would a vault pill be digested before it could do any good? Would a cell be able to take up a vault? Could vaults be toxic? Would they trigger a detrimental immune response? Just how far are we from using vaults as drugs? Why, Mr. Smith, I'm so sorry that you're not feeling well. My advice? Take 2 vaults. Call me in the morning. Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell called a vault. My executive producer, Tara, and I are trying to explain what vaults are to non-scientists. In our last video, I described the process of vault packaging and explained how even large cargo gets into the vault. Today, I want to cover 3 things. First, I want to describe what happens to vaults when they are subjected to an acidic environment, like what a vault might see in the stomach. Second, I want to tell you what happened when we added vaults to cells. Finally, I'll describe the effect of vaults on cells of the immune system. Stick around and find out if vaults will act like a pathogen and induce an immune attack. I mentioned my colleague, Hal Monbouquet, in episode 14 when I described our NERT grant. Hal is a superb chemical engineer, and he wanted to approach vaults using chemical engineering principles. 2 of Hal's graduate students, Lisa Goldsmith and Marcella Yu, wanted to work on vault structural dynamics, a fancy way of saying changes in vault shape. Biologists refer to the shape of a large molecule like a protein as its conformation. Lisa and Marcella asked the question, what is the effect of pH on vault structure? In biology, pH is a measure of the acidity or basicity of water-based solutions. So, to understand pH, you should understand what is an acid and what is a base. Acids and bases have opposite properties. Acids are chemicals that can donate hydrogen ions, and they often taste sour, like lemon juice and vinegar. Bases are chemicals that can accept hydrogen ions. They feel slippery and include baking soda and soap. pH is a logarithmic scale from 0 to 14. pH is used to measure how acidic or basic a substance is. 7 is the middle of the scale, and it represents neutral pH or pure water, neither acidic nor basic. Numbers below 7 are more acidic, and numbers above 7 are more basic. At the extremes of the pH scale, on the acidic side is 0. Here you would place battery acid. For foods, lemon juice and colas like Coke and Pepsi are about the most acidic foods, with pHs between 2 and 3. On the basic side, 14 would be drain cleaner. Ammonia and soap would be around 11 to 12, and baking soda is pH 9. Remember my drawing of a human skin cell from episode 2? In a cell, the cytoplasm operates near neutral pH, 7.2 to 7.6 to be more exact, while the lysosome pH can get down near pH 4. Mitochondria have a pH around 8. Lisa and Marcella used specialized chemical engineering techniques to examine vault conformational changes in response to pH changes. Their studies demonstrated that as the solution pH is lowered from 7 to below 4, vaults disassemble at the waist into halves. So, why is this important? This finding may be relevant to using vaults as drug delivery vehicles, because we need to know what happens to a vault when it's administered as a drug. Finding that vaults are unstable at low pH suggested that putting drugs in a vault and administering them orally in a pill might not be a good idea, as they would likely be destroyed in the very acidic environment of the stomach. However, vaults would be stable in the bloodstream. By now, you are familiar with the method we use to package proteins into vaults by tagging them with a zip code that we call int. I told you all about this in episode 14. We were interested to see if vaults could be taken up by cells. We decided to package a protein originally isolated from a fluorescent jellyfish called green fluorescent protein, or GFP, into a vault to make the vault fluorescent and thus easy to follow its delivery in a live cell using a common technique called fluorescence microscopy. We used a human cell line grown in the lab and added the green fluorescent vaults to the cell culture media and waited 1 hour before viewing the cells in a confocal microscope, which allows you to view a thin slice through the cell. This is the image of one of the live cells viewed under regular light, where you can see the whole cell. When the plasma membrane of the cell was stained with a red stain and we viewed a slice through the cell, we see the plasma membrane surrounding the cell. If we look for the green vaults, we see that some vaults are near the membrane and some are inside the cell in the cytoplasm. This dark area is the cell nucleus. We don't see the green vaults in the nucleus. If we view the red and the green fluorescence at the same time, you can plainly see that some of the vaults appear to be bound to the cell membrane. That's the yellow fluorescence that results from overlapping of the green and red signal. And some of the vaults are definitely inside. This was the first time we had evidence that suggested that vaults could be taken up by a cell. However, because the number of vaults that were getting into the cell was low, we decided to look at the uptake of vaults by specialized cells called phagocytes. These kinds of cells are part of the body's immune system. They are known for their ability to engulf or eat particles like pathogens, a fancy word for infectious agents or germs. The process of eating particles is called phagocytosis. Phagocytosis, from ancient Greek phagein, to eat, and kaitos, cell, is the process by which a cell uses its plasma membrane to engulf a particle. Phagocytosis is particularly important to cells of the immune system, where pathogens like bacteria and viruses can be taken up and digested. A phagocytic cell wraps its plasma membrane around the particle, let's say a bacteria. Once it is completely surrounded, the membrane fuses to itself and the bacteria ends up inside the cell. However, it's not actually free in the cytoplasm. It's surrounded by a piece of the cell's plasma membrane, and this structure is called a phagocytic vesicle. So, what happens next? Just walling off the bug might not be good enough. The phagocytic cell wants to kill the bacteria. To do this, it can fuse an acidic vesicle, a lysosome, with the phagocytic vesicle. The two compartments mix, and the fusion lowers the pH of the vesicle and delivers specialized enzymes that can kill the bacterium and chop the bacterial proteins into pieces. One important type of phagocytic cell is a dendritic cell, or DC. DCs are very important cells of the immune system that eat pathogens and chew up pathogen proteins into peptides. The peptides are presented to other immune cells, and an immune attack on the pathogen infection is triggered. When we added the fluorescent labeled vaults to DCs, we discovered that these cells gobbled up the vaults quickly and efficiently. They phagocytosed the vaults as well, or even better, than they ate bacteria. We also learned that when DCs ate empty vaults, they were not treated as foreign invaders. Vaults did not trigger an immune response. Finding that vaults were non-immunogenic, was an important milestone. This finding got us one step closer to being able to use vaults as therapeutic delivery vehicles. I'm Lenny Rome, the Vault Guy. Join me in my next episode where I explain some basic cancer cell biology and how we got into the cancer therapeutic area. As always, like and subscribe and please comment. You can ask questions about anything I discuss in the channel: vaults, cell biology, nanotechnology, etc. Your questions are the fuel that keeps my engine running.